Welcome back once again, everybody. This is chapter 6. We're going to be talking about microbial genetics today. Let's get on into it. Right, here are our learning outcomes. Again, for any of you who want to look into that and see what you're expected to learn from this chapter. These will be available online, these slides. Okay. Getting to talk about genetics, we're basically having a look at genetics from a bunch of different levels, not just from the organism, the cellular, the chromosomal, the molecular, breaking things down chemically into the actual nucleotides, what they are, what order they're in, and what it means, how it's read by the body, and how it serves the body. So the genome represents the total sum of the genetic material of an organism. Most of that is going to exist in the form of the chromosomes. Whether you are a bacteria or a eukaryote, it doesn't really matter. We do have the extra chromosomal or non-chromosomal bits of DNA that can exist within organisms. That can include plasmids, which are little tiny bits of extra bits of DNA, typically circular in form. And eukaryotes can have these as well. So it's not only exclusive to the bacteria. And the organelles themselves have their own DNA. So in eukaryotes, we have our organelles like the mitochondria and the chloroplast that whenever our cells divide, they have to divide themselves, and they have to, police that themselves with their own genetic signals and all of that, and duplicate their own genome because they have their own chromosome. But then whenever we are looking at the genome of like a species, for example, like us, then our genome includes counting that actual mitochondrial DNA. All right, so genomics is the term that refers to studying the organism's entire genome. So everything that would be included there. All right, so structure and function of the genome. We keep talking about chromosomes. So what are chromosomes? They're just the neat packages of DNA that they exist in. So whether or not, if we talk about in eukaryotes, we have, I did it again. We have what we are used to seeing our chromosomes laid out as, but this is like a karyotyping, where they look sort of like this, and then they'll be like next to each other, and they'll be, connected, and all of that in pairs typically. Whatever, you get the idea. So those are our chromosomes. So those are little packages of our of DNA that exist discretely. Distinctly is the word I was looking for. Okay, so eukaryotes we typically are going to occur in pairs, like ours are, one from a mother, one from a father. That's diploid, and then whenever we are going to make our gametes and they have to come together, those are haploid. So we have single chromosomes, so they can come back together and only have two, because that's how many they're supposed to have. So in pairs, anyways, that's what I'm trying to say. Bacterial chromosomes are typically circular, and they usually, some bacteria can have more than one circular chromosome, but some only have one. So just depends, but we can have quite a few chromosomes, linear chromosomes in eukaryotes. So a gene is an informational packet that typically codes for some sort of function. That function often is going to be read out as a protein as a result. So these are structural genes that do code for proteins, genes that code for RNA machinery, where we would be using for protein production. So here we're talking about the makeup of the actual ribosomes themselves. And then regulatory genes, or the transfer RNAs. We'll get into how that works soon. And then regulatory genes that actually control the gene expression. Those genes aren't going to be necessarily expressing something in particular other than a repressor or something like that, but nothing that's going to function otherwise for the cell. So the genotype refers to the genetic makeup of the genes of the organism, whereas the phenotype is what is actually being expressed. So you might carry a gene for some sort of rare trait that could be potentially, harmful to you, but since you have another copy of it from, maybe the damaged one was from your mother and you have another copy from your father, maybe, that would mean that you wouldn't even know that you were carrying that damaged form of that gene. And so that's your phenotype, because you are expressing it as though, you were normal, but you'd be carrying that gene still within your genetic makeup. So that's your genotype. So that's phenotype versus genotype. Okay, this is just talking about genome size. I don't need you guys to worry too much about this, but it is just comparing the size of like a bacteria, typical bacterium, E. coli, widely studied, widely used in microbiology. This there's going to be about 4,000 genes, and whereas humans have about 23,000 genes on their 46 chromosomes. DNA consists of the building blocks of a nucleotide, and then nucleotides themselves consist of a phosphate group, a deoxyribose sugar, and a nitrogenous base of some kind. And the nitrogenous bases, these can be the purines and the pyrimidines. And these are what determine basically that ATCG code that's going to get read and to make proteins later on. So A represents adenine, and that always is going to pair with thymine, always. And then guanine is always going to pair with cytosine. So yeah. DNA itself has an anti-parallel arrangement. This just means that, like, okay, so one side of, I'll show you a picture of it, it'll make more sense soon, but it runs in a 5' to 3' direction in one direction. So it's literally, so if it's running this way, this is 5' at the beginning and 3' at the end. And then the other strand is a complement to it. So, A goes with T and C goes with G always, etc., back and forth down the entire length. So it's complement to one another. But anyways, this is 5' over here, and this is 3' over here. So that's what it means by anti-parallel. That's just based completely on the last carbon of the sugar at the end. So let me show you. I know it's not really helpful. But this is the sugar, the deoxyribose, and they number the carbons in that. That's what chemists like to do. So the carbon atom that's at the end, at this one is going to be the 5' one, and it's going to the 3' end, and this is the 5' one, and this is the 3' end. So that's just referring to what sugar is at the end, what carbon of the sugar is at the end. That's all that it's referring to. So not exciting, and just, I feel like a sort of trivial way of talking about this, but that's what 5' to 3' means, okay? An anti-parallel. So we're going to talk about semi-conservative replication of DNA. Basically, what we're talking about with this is that if we have two strands of complementary, and then 5', and then this is 5', and this is 3, and this is 3', and whatever, and they have their little A to T and G to C makeup going on here in their little hydrogen bonds across one another, that makes up the double helix, right? So what we do is we split that apart. So this is 5 to 3. I just want to keep this going so that we keep remembering how it's set up. So this is 5 to 3. And now we have, essentially a half of a double helix and another half of a double helix. And then what can happen after that is we can come in and make copies. So this would be 3 and this would be 5 for that side. And that is 5 and that is 3, because it's always going to be anti-parallel. Then we make our little complements. We make our little complements, A to A to T and G to C. And now we have two full double strand DNA. Now, of course, this has to happen for the entire chromosome every time that the cell wants to replicate itself. So anytime you have to repair damage, anytime you get a cut, anytime you're growing, things like this, and you need new cells, they have to be able to make a copy of the entire genome, basically, not basically, but literally, in order to preserve that genetic makeup. So this is how it's going to work. And I'm going to explain it to you. I'm not going to worry about this picture, but this is essentially the setup of how this works. We're going to have kind of these origin of replication, which is just where you started. And it's going to bubble out. And then this side from wherever it started goes in towards and splits apart. It's splitting apart here. And it's just going to keep splitting and splitting and splitting and opening up that bubble wider and wider and wider. So we're going to get into the details of what exactly in the heck is going on over here and over here. The heck is this? What's happening? Why is this in little pieces? What are these arrows and whatever? So we're going to get into that. Here are our enzymes that are involved in DNA replication. Do be aware that helicase is what's going to unzip the DNA helix. Primase is going to synthesize an RNA primer because DNA polymerase has to work off of something. It can't just add on to, the ether. It has to add on to something. So the primase is what creates the primer for the DNA to work off of. The DNA polymerase, to work off of. So ligase is going to seal any little pieces together, close those gaps, those phosphodiester bonds. Topoisomerases, these guys are going to be involved in relieving tension in the coils of the DNA, because as you were to take something that is already coiled and you're basically spreading it out and opening it up, the coils on the end just get tighter and tighter and tighter, sort of like a phone cord. You probably have no idea what I'm talking about because that is a long time ago. But yeah, so you get the idea. So the topoisomerases are there to deal with the coiling of the DNA. So we already mentioned how this is semi-conservative replication, because you have an old strand and an old strand come apart, and then we have old and new strand getting made off of it, and then the other old and the new strand getting made off of it. So the new ones each consist of old and new, right? The new daughter molecules will still be identical to the parent, but one came from a template, and then the other is a copy of that template. So DNA polymerase 3, this is once we get the DNA helix unwound and we're going to start actually making the copy of all of our genome. This is going to be the job of DNA polymerase 3, adding on those nucleotides in order as we're unzipping the DNA. So that's going to use the parent strand as the template, adding on the appropriate nucleotides, A to T, G to C, can't say it enough. And it can only add, again, to an existing chain. So that's why we have to have primers there in the beginning. And it can only add in the 5' to 3' direction. So this is going to cause a problem, right? We're going to see why in a moment. So the replication fork is the actual place in the helix where the strands are unwound and that replication is actually taking place. So the circular DNA molecule will have two replication forks, kind of opposite one another on the strand. So that's for bacteria, right? We don't have circular DNA. We'll have to just have several origins of replication in our chromosomes, and they'll just be linked together by our good old friend DNA ligase. So, right, so that's what's going to be going on. We already know about RNA being our primer. So the leading strand, as we are opening up our strand, we are going to be able to go in the 5' to 3' direction on one side, only one side. And that, as that opens up the replication fork and is able to, push through and push through and push through and keep going, it just keeps adding on nucleotides in that 5' to 3' direction. However, the other side of that, so we had our parent strand, right? Which was like old strand, and now we're opening this up, and now we're making a copy on each side. Well, only on one side is it going to go just straight through. The other side, as it gets pushed open, we will do it in pieces. So it's like it gets pushed open, we make a piece of what's left over here. It gets pushed open even more, we get a copy of that piece that's over here. So now the daughter strand on that side is made in pieces because we can only go in the 5' to 3' direction, and that strand is facing in the opposite direction. And so that's why we have to make everything in pieces there. That's the lagging strand where we have to make it in pieces going in the what would be the opposite direction, but we can't go that way. We can only go 5' to 3'. So we will later on seal that together using DNA ligase. Those pieces that are made as we are going, those are called Okazaki fragments. So let's take a look at this. So we have our parent strand in the blue here. Parent strand in the blue, so we are separating everything out. So we have one parent strand over here and one parent strand over here. We can only add in the 5' to 3' direction. So we can just go straight on through on that side. That's the leading strand. However, as we go opening up, we cannot go this way. Impossible. This is against the laws of chemistry here. So that's the rule with DNA polymerase. They can only move in the 5' to 3'. So that means that the only direction that we are able to move is going to be in this direction. And that means that as this side over here, that parent strand, as this is going to open up in this direction, that is going to open up another spot for us to come in and make a copy on this, and then open up, and then copy on that, and open up in little fragments. Those are the Okazaki fragments, and we are just going to seal those together using DNA ligase. They also, each fragment has to have an RNA primer because we have to build it off of something. So these have their own RNA primers, and we have to go in there later on and add in nucleotides to replace the RNA with the DNA, because we need deoxyribose, and we need, so that's instead of ribose, right? And we can't use uracil. So that's the idea of how that works. All right. With linear DNA, so we had the two origins of replication on a circular bacterial DNA genome, but for the linear DNA, like what works for eukaryotes, this is going to be similar to what's going on in the circular DNA of the bacteria and archaea, but we're going to use some variety of DNA polymerases are involved in all of this, and we have multiple origins, not just the two. And then we have the topoisomerases that are available because the size of our chromosomes is much larger. So we need a little bit more help there. So that's the idea of how that's going to work. So the idea with the central dogma of genetics, this means that we have transcription and we have translation in that order. So that would mean going from DNA to RNA, RNA to protein. And what's really going on there is going to be DNA is like our blueprint that we keep in the Smithsonian, right? That's the original copy. You're not going to take that out of the Smithsonian if you want to look at part of it and, make a, building based on that blueprint. So no, so what you do is you make a copy of the part you're interested in, you go find the gene that you're interested in, and take that DNA and you make an RNA copy that is the messenger carrying the message for that protein from that gene, right? That's messenger RNA. So cool. So we went from DNA to RNA, the process of making RNA from the DNA blueprint. So working copy, that's a process of making that's called transcription. So we've written down, we've transcribed, and now we're going to take it out of the Smithsonian to our work site and translate it to something usable into our product that we're after all. And that is going to be a protein. So that's how that's supposed to work. That is the central dogma of the steps on involved in genetics. But of course, there are some deviations from these rules, and I'm not going to get too much into it right now. But HIV has turned our world upside down with regards to reverse transcriptase, which I'm sure you can imagine what that does. Now we can take RNA and make a DNA copy with that. Super useful tool for us. Bad deal for us whenever you're getting infected with that virus. So here's the flow of genetic information in microbes. We have our DNA, and it's our circular little chromosome here. And we can use the DNA to make, our RNA, mRNA, and our ribosomal RNA. So all of these are going to work together later on, but they originally all came from a DNA area, right? So that was encoded within the DNA. So anyway, so our ribosomes, which are made up of ribosomal RNA and a little bit of some protein, that's our machinery. That's what's going to allow us to read the message of the messenger RNA and put amino acids together to make proteins. So that is translation. So that's translation. So when we made the RNA from the DNA, that was transcription. Now we're making proteins from the RNA, that is translation. We also have these other interfering RNAs. These are going to regulate genes, and don't worry too much about the details of them right now, but this includes things like microRNA, and they literally will match up with certain areas in the genome to silence them, to turn them off so that they can no longer be functional, so that can interfere with the genome. It can also interfere with the messenger RNAs. There's different ways we can silence these, the production of proteins. So the connection between DNA and the organism's traits, the primary structure of that protein, so we talked about this whenever we're talking about chemistry, primary structure is the amino acid. That's going to determine the shape and the function of that protein. How do those amino acids interact with one another or across the way from the other guy or whatever it is? That determines how that protein is going to fold, how it's going to be shaped, and how it's going to function. So overall, proteins determine the phenotype. Whatever protein is made is what is going to be expressed, what is seen. So whatever, I have green eyes. So if I carry the gene for blue eyes, my sister has blue eyes. And if I carry the gene for blue eyes, but I'm expressing green eyes, well, that's my phenotype. But my genotype would be, the gene for green and the gene for blue, for example, whatever. I don't know, but that's an example. So that's proteins are really the ones that are going to determine the phenotype, because that's the one that's being expressed. That's what's getting made, is the actual phenotype version. All right, so proteomics is the study of the expressed proteins. So in the end, what protein you're making, that determines your phenotype. That's what proteomics is going to be studying. So remember, DNA is our main blueprint. DNA, every 3 nucleotides is a triplet. In DNA, every 3 nucleotides is a triplet. In mRNA, our messenger, which is copied of that, every 3 nucleotides is our codon. And each codon, those 3 nucleotides of the messenger RNA, are going to yield an amino acid, 1 amino acid. And each codon, depending on what it is and what it spells, A, T, C in the DNA, or A, U, G in the RNA, because remember, DNA uses thymine and RNA uses uracil, right? So keep that in mind at all times. So that these are different kinds of molecules, and it's important. They are different for a reason, because they do different things. So this is essentially how we're going to get our protein, is through these triplets and codons, yielding us individual amino acids, and how these are going to interact with one another. So essentially, our codons in our messenger RNA are going to get matched up with the appropriate anticodon on a tRNA, or transfer RNA. This is an example of a tRNA, this transfer RNA. And down here at the bottom, we have our anticodon. So that's exactly kind of what it sounds like. A goes with the U goes with the A, G goes with the C, just like the T's and the A's would, right? So we replace the T's with U's. So this will match up. It's the opposite, it's the complement to whatever our 3 nucleotides are for that codon. So it matches up. It says this is the match. And then that means because it is able to match up to that codon, it will drop off an amino acid, which can then be bound onto the building protein as we are going on and on and on and on. So that's where those amino acids are coming from. They're coming from being attached to these transfer RNAs, and how the anticodon is going to match up with the appropriate codon on the messenger RNA that we made as a copy of our DNA blueprint. And I hope you're following, okay? Because it's a lot. This is something I feel like you can learn in an entire semester. So hang in there. Okay. Whew. This is just talking about different kinds of RNA that there are and how they function within the cell. I'm not going to go over too much of these details, but definitely be aware of messenger RNA, mRNA, that's what that is, tRNA, which is transfer RNA, and ribosomal RNA, which is rRNA. So those guys are the big three that you definitely need to be aware of. It's nice to understand a little bit about microRNA, but I don't know. You know it exists. I don't know. Okay, so so differences in the RNA structure. So basically, this is a single-stranded molecule that can exist in a helical form, which we saw a picture of it not too long ago. There it is. You see how it wants to be in a helix kind of over here. So it's folded back on itself and created a sort of helix back here. That's how RNA likes to exist. So it is single-stranded, technically it just folds back in on itself a lot of the time. So that can allow for it to establish secondary and tertiary levels of complexity or shape, which allows for different function as well. It does have uracil instead of thymine, U instead of T. No T in RNA. So anytime you're thinking a T, you put a U, right? The sugar that's involved in RNA is also ribose. It is not deoxyribose. So those are the big differences that are involved. Messenger RNA is a transcript of those structural genes, the ones that we're interested in making the proteins from. And then we make the RNA, the messenger RNA, in a process that's very similar to how we would replicate the DNA. And then those codons, those 3 nucleotide sets within the messenger RNA holds the actual message for the protein that we're going to make when the ribosome and the transfer RNA come and read it, right? So that transfer RNA, remember, it's going to have complementary sections that match up with the messenger RNA. And when it does, that's going to signal that that's the amino acid that's going to that codon codes for. So that codon, if it was AAA, the anticodon would be UUU, and then because of the AAA matching up with the UUU on that association with the transfer RNA, now we can have our appropriate amino acid like phenylalanine or whatever it is, and that can get added on in the appropriate sequence while we're building our protein together. So the ribosome itself is a whole lot of long RNA molecule and protein complex. That's essentially what it is. And it has size and it has a shape and everything that I'm not particularly interested in. And I'm not so sure that I'm that interested in you knowing it, except that the size of it is measured in as a weight during centrifugation. So you're spinning it really fast, and based on how it settles in a specific medium, they determine that it has a 70 Svedberg size. So whatever. So that's for bacteria. We have in eukaryotes an 80 S ribosome. So moving on, who cares? Transcription has essentially 3 steps: initiation, elongation, and termination. Really easy stuff. All right. Initiation, this is going to be started whenever RNA polymerase recognizes DNA in a region called the promoter region. RNA comes in and associates in that promoter region and just starts cranking out in a 5' to 3' direction the messenger RNA that is complementary to the actual template strand. The coding strand of DNA for that message, that would be the actual what your messenger RNA actually reads, because we're making the complement to this template strand. So that's what the coding strand is. It just contains the actual message. Anyways, so that's how that's going to be going on. We're just going to start adding on. So this initiation, we have the binding of the RNA polymerase to the promoter region. Big important concept there, that promoter. Okay, big deal. You can even set up some genetic experiments that will allow you to trigger association of RNA polymerase to certain promoter regions. And that when that happens, you can cause your bacteria or whatever you've engineered to produce a protein of interest associated with that. So that's a useful tool that we can talk about a little bit in the next, well, not next chapter, but the chapter after that, the genetic engineering one. Okay. So, right, so we're going to elongate in the 5' to 3' direction, just adding on the appropriate nucleotides. In this case, we are talking about the RNA nucleotides, so ribonucleotides with ribose and uracil, and that's what we're going to be adding. And then eventually we get to a point where we will terminate. It's basically a stop sequence where we will disassociate from the actual RNA polymerase will disassociate when it gets to that little stop sequence. And that's pretty much all there is to it. It's not really meant to be too complicated here. So our initiation, we associate at the promoter, then we elongate in the 5' to 3' direction, and then we terminate at the stop. That's not hard, right? So we have our single-stranded messenger RNA as a result. Okay. Our messenger RNA, again, is going to be read in these groups of three nucleotides that we call a codon. Basically, there's almost a pseudo-universal code for all of this stuff. And here it is. So here we have, again, if your codon reads UUU, then you code for phenylalanine. Let's say, hypothetically, you just, okay, codon reads for AGG. So then you would go to A as your first base, which is here, A, and then you want G as your second base. So you go over to this section, and an AGG is here, and that would code for arginine and whatever. So that's how you figure out how that table is set up. And you can see there's a lot of redundancy in here. So if we have any of these four guys here, AC anything, that codes for threonine, and then GC anything, that codes for alanine. So there's a lot of redundancy here, and there's a reason for that. That is what we call the wobble effect, that third codon, considering that to be the less important one. The third nucleotide is the less important of those three nucleotides in the codon, is what I'm trying to say. So, and that wobble effect is the fact that we can accommodate different, nucleotides in that third spot. And that helps preserve the genetic outcome. So the outcome of the genetic makeup of an organism, even in the face of mutations. So that's why that redundancy is there. So only the first two nucleotides are the more important ones, right? The third one doesn't change most of its sense, and sense is the actual product of the amino acid that's coming out in the end. So it permits variation due to mutation. That's one way that organisms are able to control, protect themselves from mutation and detrimental effects of mutation. So we have the DNA triplets, and we have the coding strand and the template strand. We make a copy of the template strand that happens to read the same thing as the coding strand for our mRNA. So here's our mRNA. Those now are not triplets. Those are now codons. The codons will match up with their appropriate match for their tRNA anticodon. And then those are associated with a specific amino acid. And so this is going to determine, and we'll link those together. So methionine is almost always the start. And then we have leucine and threonine and whatever. So here we have two different, we have a UG and a UGC, but both of those are going to code for threonine, again, because of that wobble effect, that redundancy in the code. So anyways, we'll keep going on and on and on and adding on our amino acids as we are reading, the codons during translation. So that's kind of how that's all set up. So we need to have the messenger RNA, the transfer RNA, amino acids, and the ribosome. So this is everything that we need in order for translation to occur. And translation has the same steps as transcription: initiation, elongation, and termination. So we always start, the start codon is always AUG. Typically encodes methionine or formyl methionine, which is just a formyl group on the front of it. So it's not, I don't know, it's not particularly exciting. It's always the same one, but we can clip that off later if we don't need it as part of the protein, but it does always start with that. And then at the end, we have a stop codon. A stop codon, the reason that the ribosome will disassociate and no longer try to read the messenger RNA when getting to that stop codon is because stop codons don't stand for anything. There's no amino acid for it. It's a stop. It's nonsense. Codon is literally what we call it because it doesn't give us an amino acid. So nonsense ones are the stop ones, and it causes the ribosome to dissociate and say that we are done. And then translocation is the process of shifting that ribosome down as we are reading our codons. So here are some step-by-step processes that I guess we can go through in a little bit of detail. I don't want to go too much words after we've already described this, but I like the pictures involved here. So here we have our start. That blue is not a useful color for this. I apologize. Let me get a different color. Go back to red. Okay, so we have our start, which is our AUG. So what's the anticodon going to be? UAC, right? So we can see that clearly there, that AUG on our messenger RNA and the UAC on the transfer RNA. That transfer RNA is carrying on its cute little self formyl methionine, F-methionine. And then, so the next one comes into the next pocket, the A pocket, and that's going to be for leucine. So that is the CUG. That matches up with the GAC anticodon. And that's going to, again, call for leucine as the amino acid. And so what happens next is we have a formation of a peptide bond between those two amino acids. And when that peptide bond joins those two amino acids together, that can allow for this empty transfer RNA to move into the E, which is the exit spot, okay? Because it doesn't need to be associated with that messenger. It doesn't need to be associated with that protein anymore or with reading the messenger RNA anymore because we've already done our job. We brought in our amino acid and it got linked on to the next amino acid. We don't need you anymore. So we kick out that transfer RNA. Okay. Next, we shift the ribosome over. That's what this weird thing is going on here. Shift the whole thing over. Then we can put that number 2 one into the P spot, which is like the peptide bond spot. And then we'll put a new one into A, and then A, it's going to have its matching little codon, anticodon situation again, like down here. It was CCG, anticodon is GGC, and we are carrying our little alanine or whatever the heck, this is proline, I guess for this one, proline, it says proline up here. So whatever, so we have proline and we're going to create a peptide bond here. Cool. So now we're associated onto that guy and we can kick out the second guy and we can have another guy come into the spot and so on and so forth. So then this guy comes in and is able to join its amino acid on, and then we will kick out that transfer RNA, and then we can add on another one, another amino acid, and then kick out the other transfer RNA, and so on and so forth until we've kicked out, until we've reached our stop codon and there are no more amino acids to associate with. And then that will cause the whole ribosome to say we done. Okay, so post-translational modifications, this just means so we can clip off the 4-methionine if we don't want it. We don't need it for the structure of our protein for whatever, but it just always has to start with that because that's just how it goes. We might add some cofactors that allow, like vitamins, that allow the actual protein to do its job. And sometimes we have other polypeptides that we can join with in order to create the functional protein, right? So, and we can have additions of, sugar group or lipid groups or whatever it is. So, yeah, in bacteria, they've got this special cool little trick that they don't have a nucleus, right? So we have a nucleus. So our transcription, our DNA to messenger RNA, we have to go into the Smithsonian. But imagine that instead of the Smithsonian, now we just have all of our archives just out under a big old tent. So people can just come in and make copies of things and start working on it right there at the site of the blueprint. So that's what happens in bacteria. So we have our actual DNA over here. So that's what this is. That's our DNA, and we are making our messenger RNA. And while it is being made, as we're adding on, so this is the beginning of it. This is where we started, and what's going to spit out in the direction building on in that way. But we can actually start translating while it's still transcribing, co-transcriptional translation. So that's what that means. While transcription is happening, we can translate. So that's what's happening there. And then we can, we can have a whole big line of ribosomes as well, working on the same thing all at once. Anyways, and then it moves on down and spits out the protein while you're actually still making your messenger RNA. So that's the idea here. They can do that. That is not something that we can do because we have a nucleus that is surrounded by a membrane. So we have to get all that stuff out of there in order to work on it. So with COVID-19 vaccines, those were mRNA vaccines, you'll remember. We would inject it into the muscle cells where it would be ejected into would actually start to make the viral protein and putting in the virus protein on the surface of the muscle cells themselves. And then our immune system would respond to that. And then whenever that happened, it creates a protective antibody against that protein that we were expressing. And that should be the protein that is on the surface of the COVID virus. And that's how the mRNA vaccines for COVID work. So it's pretty neat. So we can only have co-transcriptional translation in bacteria and archaea. We can't do that in eukaryotes. Eukaryotes, their mRNA only codes for one protein, whereas bacterial mRNAs, they can have multiple proteins coded within that one messenger RNA. But eukaryotes also have this other stupid thing called introns and exons. Exons are the ones that are going to actually code for a thing. Introns are just some extra stuff that need to get cut out. We don't need it. So what happens is we have our DNA template, we make our messenger RNA, and the spliceosomes come along and cut out the introns so that we can join the exons together and make readable form. So bacteria don't do that. They don't have that. So that's basically what the spliceosome does. It loops that intron and cuts it out so we can have a workable messenger RNA. Right. So for bacteria, they have these things called operons, and these are only bacteria and archaea. Okay, they can be inducible or repressible, and it's going to be dependent on their environment of the cell. So this will be usually associated, inducible operons are usually associated with making proteins that help metabolize nutrients. Operon will be turned on or induced when the nutrient is present. All right, so inducible operons, basically, these are going to be turned on or induced whenever a nutrient is present and we need to be able to metabolize it. So repressible operons, these guys are going to be always on, and then when there's too much product that they made, then they're going to be turned off so that it's going to be repressed. for the lac operon. So essentially we have the promoter region here. So that's going to be the part where the RNA polymerase is going to associate. Then we have the operator. So this is going to be where this is our on-off switch for transcription of this whole set of genes. So it's 3 genes. That's our structural locus. 3 genes associated with catabolizing or breaking down lactose. Okay, so cool. So the repressor protein is allosteric, and that means that it has 2 binding sites, one for the operator sequence on DNA and another for lactose. And in the absence of lactose, the repressor will bind to the operator, blocks transcription so it can't happen. And then what happens is when lactose is there, it allows for that change in that repressor so that we can actually keep going. So basically, here's a picture of it. So here is our repressor. Our repressor is locked on to the operator, so our RNA polymerase cannot go on. So then we have lactose added. It changes the shape of that repressor, and so it goes away. And now our RNA polymerase is active and can make our mRNA through transcription, and then translation allows us to make the proteins that are associated with breaking down lactose. And then essentially, whenever there's no more lactose will eventually dissociate, so it can all be broken down. So there's not enough lactose to cause that repressor to be unlocked, from that site. Then it'll turn back to its locked form, essentially, and then stick back onto that operator. So that's how the lac operon works. Repressible operons, essentially, these are only going to be turned off whenever we don't need that nutrient anymore. So like making, I think the example that we're going to have, okay, good. We have a pretty good example. Okay, so our nutrient that we're trying to make serves as a co-repressor to block the action of the operon eventually when we have too much of it. So essentially here is our always turned on sequence here, our operon. Whoa, I didn't know that could do that. Associating our RNA polymerase, it's turned on. We're going to transcribe. We're going to make our messenger RNA. We are always doing this. It's always so fun. Here's our arginine. We're making arginine. And isn't this fun? And we're producing arginine. And here it is. And whatever. And we have eventually so much arginine that our repressor protein, once it is activated by arginine, will bind on to that site on the operator so that we can no longer do transcribing. Because there's enough arginine, we don't need any more, stop making it. So that is how we repress that one. Right. So there's all sorts of different ways that we can regulate genes as far as expressing these genes in our genome. For eukaryotes, this is usually in response to our own environmental stimuli, but we just aren't going to have that multi-protein situation that we see in operons for like bacteria. But yeah, there's a lot of different ways that we can block transcription, whether it is with like things like microRNA or things like here we are saying DNA knot. This DNA knot where our cytosines allow a bind to other cytosines to form a knot in the helix of the DNA to block the promoter so that we can no longer, do transcription of that. So we can literally physically block it that way. So it's just one example, but there's a lot of different ways that can happen for eukaryotes. Okay. Anytime that, okay, so this is just talking about bacteria that can turn on or off genes that can lead to phenotypic changes, so actual expression of certain aspects of the bacteria. These are heritable, and their ability to turn it on and off is heritable as well. So that's what that is. So recombination is going to be talking about whenever we have genes from one organism, and then we're going to put it into another organism, essentially. So that's what DNA recombination is. We're going to talk a lot more about this in the genetic engineering chapter, but we're going to get into this just a little bit now. But so plasmids are particularly useful as those extra chromosomal DNA. We can use those as a means for us to insert genes into the plasmids and then control expression of those genes in those plasmids that way. All right, so horizontal genetic transfer of genes. This is going to be something that's done by bacteria, not by eukaryotes. That's anytime we have genes being passed from organism to organism that are not related. We are not dividing and new ones have our genes. No. We're talking about like an E. coli and an E. coli that are coming up near one another and literally transferring genes to one another. That's horizontal gene transfer. Yes, so we can have some, okay, fair enough, some horizontal gene transfer that would be mediated by viruses, but we don't have the means to do this intentionally, really. So plasmids, we already talked about this. Circular DNA has extra genes. They're not necessary for survival. That's important. They're not necessary for survival, but they can have useful traits like antibiotic resistance. Then we also have conjugation, that is just forming a bridge between two cells, which this is going to go over, and that can allow for us to make a copy of a plasmid or a bit of DNA and then pass it into the next cell. So that's what this is depicting here. I don't need you to know about the details of F factor versus HFR transfer. No, just know that conjugation is going to be involved with passing DNA from cell to cell through the sex pilus. So that's the connection that is made between those cells. All right. Yeah, so there's all sorts of genes that can be typically associated with this, things like antibiotic resistance, resistance to other drugs. Yeah. Transformation, that's just picking up DNA from the outside environment. You have to be a competent cell. That just means cells that are able to accept that genetic material. And if you are competent and able to do that, it would be DNA that would be in the outside environment, maybe from dead cells that are surrounding. And you can pick up those bits of DNA and they can become incorporated into your own genome. A pretty interesting little experiment that was done by Frederick Griffith. We had Streptococcus pneumoniae capsule that had capsules and didn't have capsules. So capsules, again, that's going to be a virulence factor. It's going to make things more effective at being, bad for you. So the guys that had the capsule were causing the mice to die. The guys that didn't, the mice were fine. So they killed the capsule guys, mixed the dead capsule guys with the live non-capsule guys. And then the non-capsule guys now can make capsules and cause mice to die. Then it was shown that seems to be due to transformation because the non-capsule guys picked up the DNA of the capsule guys that they were mixed with. So that's how that worked. Pretty cute little experiment. So transduction. This is, it says the case of piggyback DNA. Essentially, you get infected by a virus, and that virus can take with it different bits of DNA, either from you to the next host or from a previous host and then put it into you. It could be predictable genes, or it can be any gene anywhere in the genome, depending on the two kinds of transduction, which are generalized and specialized. In generalized, we have random fragments that'll get packaged up with the viral genome, with the viral particles. So those random pieces of DNA from the first host get packaged into the new viral particle, and then it will transfer that into the next host cell when it's infecting it. And then that can transfer genes. So that can transmit any gene from the bacteria, one bacteria to the next. It's unpredictable though. With specialized transduction, we have our incorporation of our genome as bacteria, incorporation of our genome as bacteriophages, as viruses that infect bacteria. That these guys come along and insert their DNA into the host genome, to the host chromosome, and that will be often at specific spots in the host chromosome. And then whenever they, so that's going into lysogeny. And then whenever we go back into the lytic phase for making more viruses, they can take out the gene from that host with them and then move on to the next host and give them that gene associated from where they were hiding in the genome of the previous one. So that's how that works. Transposable elements, I'm not too particularly interested about these, but these are jumping genes. They can move from one part of the genome to other parts of the genome. So from plasmids to chromosomes, chromosomes to plasmids, and et cetera. These are just jumping genes. And these can occur in eukaryotes and in bacteria. So these can end up scrambling the genetic language. It can be, useful, but it could also be harmful. Of course, it can cause weird mutations, weird splicing of the DNA, changes in morphology, often in pigment, like what we see in multicolored corn. Transfer of drug resistance between bacteria is also a possibility as a result of transposons. So those jumping genes. A mutation is any change to the nucleotide sequence of the genome at all from what it should be, the wild type. So if it deviates from that, it's a mutation. You can often see this expressed in phenotypes, but not always, right? So wild type is the most common natural form of something, whereas a mutant strain is the one that mutated from the wild type. There's different ways you can get mutations, right? Spontaneous mutation, we have random changes in the DNA arising from errors that occur randomly just from replication and it not being perfect. Unfortunately, it's just not always perfect. Then we get induced mutations. So being exposed to mutagens, anything that causes mutation or is likely to cause mutation, like radiation or, certain chemical agents can cause mutations. Point mutations refer to a single base being changed. So an A having mutation into like a T. So just that one base pair gets changed. Lethal mutations are any mutation that causes cell death or death of the organism. A neutral mutation has no whatever. It's a mutation, sure. We can see that it changed the amino acid sequence maybe of the actual outcome of the protein of that gene. So cool. So that maybe that happens. But if it doesn't change the phenotype at all of the organism, or doesn't create really any beneficial or harmful changes, that's a neutral mutation. So we can break them down based on the actual result at the amino acid level. A missense mutation is anytime that you have a change in that codon, like the AAA, anytime that's going to be changed to cause a different amino acid sequence. So instead of alanine, now we have phenylalanine. So if that change were to happen, that would be a missense mutation. A nonsense mutation is whenever you have your codon, like AAA, and it gets changed into a stop codon. So that's a nonsense mutation. So there's no more amino acid there. And that can be fatal, of course, right? So if that happens early on in the protein and we have just that one little change, that can cause severe damage to that protein and potentially cause harm to the organism. A silent mutation, remember how we have that wobble effect? So you get like the first two for a lot of those, the first two nucleotides of the codon are what's important. So if we change our base, our nucleotide, but the amino acid doesn't change, so same one, both same amino acid results, that's the silent mutation. And then a back mutation is whenever you have a mutation from the wild type, and now you're mutating again back to the original wild type. All right, so this is just looking at some of the mutations. That the top is the actual normal gene. This is the DNA sequence, and this is the protein result. So in a missense mutation, we had methionine, threonine, and aspartate, but here we have glutamate, and that is because we changed from CTG to CTT in that. So that would be GAA in the RNA, and that now will code for glutamate instead of aspartate. So that's an example of a missense. So we could go through the other ones as well, but there's other ways we can screw up your genetics, basically. So the frame shift mutation this is going to be due to an insertion or deletion of a nucleotide, usually one, maybe two. But if you do it in a multiple of three, that's not going to disturb the reading frame. It's going to stay in the same reading frame, essentially. So if we alter the frame of the messenger RNA, that will result almost always in a non-functional protein. So that's going to change every single amino acid from that point of the mutation down field. So this is an example of that. We have deleted this G right here. And so now we have the C, and we've shifted everything down all the way up. So now instead of TGG, we have TGC and whatever. So you see how this affects the actual RNA and the actual codons that are going to be read. And so now instead of reading the correct codons and getting the correct amino acids, we've shifted our entire reading frame, and now we're going to be making these instead. And instead of having amino acids and keeping going, we have the introduction of this stop codon that's going to dissociate and create a disastrous effect for that particular organism, right? And so I guess this is just talking about other frame shifts and things like that that can cause effects as well. So anyways, so ways that we deal with mutations. Photoactivation is a way that we repair light damage caused by ultraviolet radiation. Essentially, ultraviolet radiation causes the T's that are next to each other in the DNA to bind to each other, next to each other, instead of across to the A's across from them. So they're not behaving correctly. So that can be the damage that's done by ultraviolet radiation. And we can repair that using photoactivation, where we have visible light that activates enzymes that will go in and literally cut out the bad pieces and make new ones to repair it. But there's also mismatch repair, where if we're going to be going through the DNA and we see that it is mismatched, it's not matching up the way that it should, A isn't binding with T or G isn't binding with C or whatever, we can also repair that as well if we catch it at the right time. So this is excision repair, which is what we would be doing for that, where we're actually cutting out those, they don't actually match up the way that they should. So you see how these shapes don't bind to one another. That's not the right thing that goes there. So we just cut it out, and we can put in new ones that are going to match here. And the enzymes that are involved in that allow us to cut out the bad and put in the correct ones. It's mismatch repair. Again, you have to do that before certain points in the cell cycle. So this is the Ames test. This is how we detect carcinogenic potential, cancer causing or mutagenic causing potential of a chemical. So it's a little bit of an issue explaining this, but I'm gonna try my best for it. So the culture of salmonella, and it is not going to be able to make histidine. So we plate it onto a plate lacking histidine and a test chemical. So this organism can't make its own histidine. So if we don't provide it with histidine, it does not grow well. Okay, so that's what this is saying. So our control plate is showing us that when we plate that I cannot make histidine organism onto a plate without histidine, this is what it should look like normally. Okay, now we've got our test plate. We've got no histidine added to this. Okay, so it still cannot make histidine. No histidine, none. Okay, still can't make it, not on our own. And we've added our chemical that we think is cancerous to the medium in here, into the pink plate. And now we will plate our unable to make histidine salmonella onto this plate and then let it grow. And if we get a lot of growth, that means that our bacteria have mutated so that they can now make their own histidine again. So that's bad. So that means that this would be a mutagenic chemical that we've added to that media. So that's how that's supposed to be working. And how we can look at certain chemicals to see the safety based on that. So a lot of times whenever we have mutations, a lot of times they're not repaired. I mean, they're usually just left and they just don't get repaired in time, or they can even get passed on, especially when talking about bacteria, they can get passed on to other bacteria. Now they've introduced mutations into their system, and etc. So some of them aren't repaired, and the effects depend on the mutation and depends on the organism and how it lives in its life. Mutations are permanent and heritable sometimes whenever we're talking about the ones that are going to affect especially your gametes. But in bacteria, they don't have gametes, right? They don't have sperm or egg. So they have to, any mutation they have in their chromosome is heritable to the next, group. That's just how it works. They're just too tiny. So anytime that happens, we have passing on to our offspring, right? A small number of mutations can lead to success. So mutations can lead to drug resistance and stuff like that. And the ones that have those mutations are going to be the ones that survive and pass on the mutations to the next, offspring. So that's great for bacteria, not so great for us when that happens, of course. And that can happen just randomly. Mutations that allow for drug resistance can happen randomly. We can also have those conjugation or transformation or transduction that would allow for trade or introduction of drug resistance as well. But you give them enough time and bacteria will become resistant. All right, so here is your question of the chapter. I'll let you guys think through this one. What are some ways that bacteria may be able to develop antibiotic resistance? I did just mention that just now. And how would they transmit this to other bacterial cells? So actually go through that and answer that for yourself so you can understand it. And I will see you guys next time. This was a long one. I know, sorry.